Okay, so Wernicke's region, just a short reminder, we've got area 41, primary auditory cortex, 42, Wernicke's area in the classical sense is actually only the posterior part, or was described as only affecting the posterior part of area 22. But, well, we've got different patients with different degrees of lesions. Okay, so with Wernicke's area, I want to start with the auditory region. And this article here by Rademacher is the one that was actually mentioned yesterday evening concerning the volumes of primary auditory cortex. And they examined a total of 27 brains. They examined the left and the right hemisphere, and so approximately half of them were male, half of them were female, nicely matched. And what you see here is a drawing of a brain. So you've got the left hemisphere here and the right hemisphere here, and all the lateral prefrontal cortex, medial, the cingulate cortex above the temporal pole, everything that was above the lateral fissure has been cut away so that you can look from above at the dorsal surface of the temporal lobe. And in this study, Rademacher and colleagues, they looked at variability in the macroscopical landmarks of the auditory region. So they identified the transverse gyrus, also known as Heschel gyrus, and they saw that there was only one Heschel gyrus in 70% of the examined hemispheres, and there were two in 24%. That's an example here. So here in this right hemisphere, that's an example of two transverse gyri, and then three were only present in 6% of the examined brains. Now, of course, you can't have a gyrus without sulci delimiting it, so they also looked at those. Now, obviously they found that the first transverse sulcus, which is labeled here in blue in the drawing, was present in all brains, and Heschel sulcus, the red line, was also present in all examined brains, which isn't surprising since all brains had at least one Heschel gyrus, and these two sulci are its borders. But then the intermediate sulcus, which is the one that would separate the two transverse gyri when present, was only present in 41% of the cases. Now, the interesting thing about this study was that when they compared this variability and they looked for left-right differences or differences between the male and the female brains, they found no differences whatsoever, okay? No significant differences. And, well, this, I suppose, isn't surprising since normally both our ears receive the same amount of information, and it's not that our right ear is more tuned to, I don't know, numbers, for example, and the left one to music or language or whatever. Okay. When we look at the cytoarchitecture of this region here, what we see is a picture like this. So this is a cell body stained section, a coronal section through the, that is the Heschel gyrus, so the transverse gyrus, and that there would be the superior temporal gyrus. Here we've got the superior temporal sulcus, and this here would be the lateral fissure. And Patricia Morazan, this was her PhD. She examined the primary auditory cortex. So I'm going to label Brodmann's areas on here. for you so that you can find a correlation between these arguably strange names and the Brodmann numbers, which are the ones that are more widely used in the literature. And the reason why at the Institute this designation was brought in was because Brodmann defined cytoarchitectonical areas, and he just numbered them based on which one he came up to now. So there is no apparent logical sequence in the position of the areas and the numbers. And these areas were then used to label activation sites elicited by tasks and functional imaging studies. And then people started using names in a way. Well, you've got the primary visual cortex, V1, which was identified functionally, and then you've got Brodmann's area 17. Okay, so those two, there's a good overlap. But then you've got other areas such as Brodmann area 18 go into that, and then you don't have a single functional blob for that, and then people started using the wildest names. So the idea here was to use a neutral nomenclature without any kind of hint as to what function it means, because we're trying to say, okay, this identifies a cytoarchitectonical area. That was the main region. And then the names come from where we are in the brain. So TE means it's the temporal lobe, and there's OC for the occipital lobe, and FP for the frontal pole, and so on. And then they're just different numbers to identify the different areas. And if you use this TE 1.1, 1.0, it's, this begins to get then. It does begin to get complicated, and it borders on your question earlier on as to what is an area exactly. And the thing is that these TE1 areas, these different subdivisions, they all have a very, very prominent layer 4, which is indicative of a primary sensory area. But then they do have differences in laminar patterns in other layers that led to these subdivisions in 1.0, as I say, 1.0, 1.1. Here we have TI would be temporal insular area. So basically the TE1 areas would correspond with Brodmann's area 41. The TE2 areas, all lumped together, would be Brodmann's area 42, and then TE3 and TE4 are a part of Brodmann's area 22. So Brodmann's area 22 can be divided into at least 2 areas, but we haven't finished doing that. So that's why, basically that's why there is no outer border here of TE4, because that hasn't been mapped yet. And then to bring this image here back into the dimension of what does it look like on the surface of the brain, I've color coded them again here. So red would be Brodmann's area 41. It's basically located on the transverse gyrus, but not restricted to it necessarily. Then we've got the temporal insular area here. Caudally on the planum temporale, we've got TE22, and then TE3 goes onto the surface and moves into the superior temporal sulcus. Back to the question, correlation between cytoarchitectonical borders and macroscopical landmarks. That is depicted in this figure here, also from Rademacher's work, because he, in this article, Patricia collaborated and she identified then TE. So what you see here are serial sections through basically, well, this is the superior temporal gyrus and Heschel gyrus. This here is the superior temporal sulcus, and we've got 6 different brains, 6 brains. The sections here is like at the bottom part of each group of sections. These are located rostrally, and when you move up in this series of sections, you're moving caudally along the temporal pole. So and what you can see here is that area TE1, which is this black labeled area on the brain and along the cortex, it's not only found on the Heschel gyrus because, well, okay, so it does start here, but there is no Heschel gyrus here. It's already finished. Here we've got Heschel gyrus, but then we're moving onto the neighboring, onto the planum temporale. So for the primary auditory cortex, again, there you can't say it's restricted to Heschel's gyrus. Okay. Moving back to the cytoarchitecture of these areas, I can answer your question earlier on. The main difference between the TE1, TE2 and TE3 regions is found, well, the 2 main differences. The first one is found in layer 4. So if you compare area 4 here in TE1 with that of TE2 and TE3, you'll see that layer 4 in TE3 is much narrower than in TE1. And if you compare these two, what you see is that actually cell density is higher in layer 4 of TE1 than in that of TE2. So this is showing us that basically the main function of TE1 would be receiving thalamocortical input, which is the main function of, or it's the target. Layer 4 is the target of thalamocortical input, so you've got the sensory signal coming in via the thalamus to primary, in this case primary auditory cortex. The same actually holds through for primary somatosensory and visual. Sorry.
Is it true for area TE1 that it gets many more projections from other cortical areas? Typically there's way more top-down projection than bottom-up projection. So is that true for TE1?
It has a very wide layer 3, which is one of the basic, it's the basic layer for cortical-cortical communication, if you want to say it that way. There are other areas in the brain, for example, that have a much thinner layer 3. So the fact that TE1 has a relatively wide layer 3 is telling you that cortical connectivity is also important for this area. So it's not that it's only interested in the information that's coming up from the thalamus and forget the rest. It has to pass that information on to the other areas, and it also it receives input.
I guess also that's quite a big layer 5, so basically you send out auricular, like corticothalamic projections as well, so they will complement the sort of thick layer 3.
And not just corticothalamic, but there's also cortical connectivity. Yeah.
Layer 5 and also the back projection to the layer 6 and 1 from the
So you see, I don't have a slide showing that, but TE1 is one of the areas that is relatively thick in the brain, meaning that it has a thick layer 4, but it also has a thick layer 3 and a relatively thick layer 5. So it is integrating input that it receives from other areas. Okay, but the difference, or another difference, between TE1, TE2, and TE3 is the fact that you have large pyramids. You have a differentiation within layer 3, which becomes more apparent when you move from TE1 up to TE3. So TE3 is what we'd call a multimodal integration area, and that is one of its hallmarks is this differentiated layer 3 and the large pyramids in the deeper layer 3. Meaning that for this area here, cortical input is more important than it is for TE1. So you can't say that it's not important for TE1, but this area here receives more input, more information via layer 3 than it does via layer 4. Okay?
Do we have any sort of sanity check to verify that indeed the width of layer 3 is a good indicator of how well connected that region is? So for example, if we look at the thalamus.
Not in humans. Not in humans. Because tracer studies, real hardcore tracer studies, can only be carried out in animals. But you do have this kind of studies in rodent brains, in primate brains. And that's why we know that layer 3 is the layer from which cortical connectivity or most cortical connectivity originates and where they end, and we know that layer 4 is the input layer, target layer for thalamocortical connectivity, and layer 5 is the one with axons, with cells sending their axons down to other cortical regions as well, but basically subcortical structures.
So these studies show that in the thalamus itself, these layers are proportionately large as proportion to the amount of connectivity that the thalamus has then, I guess.
That areas that are involved in multimodal areas tend to have a wider 3 than unimodal areas.
Now, if you compare between areas here, because should we also take into consideration the volume, because it can still be the case that the volume is, so we have no surface area. But that doesn't necessarily mean that the volume in that particular total area is
relative size of each layer compared to the...
Exactly, to the total. In different parts of the brain, like primary visual cortex is surprisingly thin compared to the auditory cortex. It's because it's got an impressive layer 4, it's got a relatively inconspicuous layer 3, whereas well, here.
Is the total volume of area TE1 comparable to TE2?
Oh, no, I wouldn't talk about volume in that sense, because V1, of course, is the whole calcarine sulcus, which is enormous. Now, one thing is surface. So the surface of V1 would be larger than the surface of TE1, but if you're looking at cortical depth or cortical width, so the distance between the surface and the white matter, that is larger in TE1 than it is in V1. So then, of course, your volume is yeah.
So I guess to measure the sort of size, length of layer 3, in this case, you have to do relative to the whole length. Yes, of course. When we talk about a wider or a narrower layer 3, it's always relative to the total depth of that area.
So you just mentioned that so in primary visual cortex, layer 4 is much more impressive.
In the primary visual cortex, layer 4, this is again, you know, we're talking about different areas and do you have areas and sub-areas and so on. This is where I laugh at cortical layers. So the cortex or neocortex has 6 layers. But if you go to the primary visual cortex, so you've got a layer 1 and a 2, and then you've got a 3, but actually you can divide 3 into A and B, but then you've got a 4A and a B and a C, and you've got an alpha and a beta as well, so it's A, B, C. You've got 5 sub-layers total within layer, what's layer 4 in here.
The reason I'm asking you because there's, you know, the tendency to want to make analogies between primary auditory cortex and primary visual cortex, and then clearly fails. And it's interesting that it fails on anatomic grounds because primary, you know, auditory cortex gets its input from an area that's already done all the heavy lifting, namely the inferior colliculus. Right? So by the time you get to primary auditory cortex, you're actually a very pre-processed signal. And the kind of stuff that has to get done in primary visual cortex, you know, more or less equivalent, is just done, right? Because the brains, you know, the brainstem nuclei on the auditory system they do a ton of the basic pre-processing.
Yes.
IT does all this stuff. So it's got to be a different kind of area. So the functional equivalences, I think, are very dodgy to make. And it's interesting that you find so you know, direct a difference there. I think that's
in the structure.
Yeah.
You see, that's why structure is important, and that's why structure counts, and you can't have a function without the structure. And I've lost my line. I might remember it later, and we can talk about during the coffee. It's difference in auditory and visual. Okay, so they're both basically the only thing they have in common is that they're both primary, or we consider them primary, but that's also a too simplistic way of viewing their function actually, because that would mean that the only thing that. Well, I know, but
it's still a riddle, because you take away the auditory input and then auditory cortex, these areas become involved in peripheral vision. You take away input to the visual cortex and all of a sudden they do verbal memory or tactile processing. So if the function structure mapping is so direct, I wouldn't be able to understand why it can take up a completely different functionality.
But that's almost a philosophical question. Okay, let me move away from the primary auditory cortex into the planum temporale and part of Brodmann's area 22. And we mapped this with receptors. Now this is a very noisy slide. It's because I'm showing basically results from 2 different studies here. So they're the borders that Patricia found on the cytoarchitectonical sections, the cell body same sections. She could replicate all of these borders with receptors. And then went on to parcellate, so TE2, which cytoarchitectonically she'd only found a TE2, and with receptors she found a subdivision TE2.1 and 2.2, for example. And you wanted to know if receptors, if they always if we can group them into different kinds of receptors.
And if they show sort of complementary distribution. So
well,
if you group them, they will kind of be more homogeneous.
That doesn't happen with receptors for a single. So we study different receptors for different neurotransmitter systems, right? So glutamate can bind to AMPA, kainate, NMDA receptors, and and so on.
If you collapse the glutamate binding.
If you collapse the glutamate binding, what you find here, for example, on the Heschel gyrus is that AMPA receptors are more highly, or the higher densities on the superficial layers than in the deeper layers. Kainate receptors, you find the opposite pattern. Kainate receptors are higher in the deeper layers than the upper layers. Now, the interesting thing is that binding of glutamate to the AMPA receptors doesn't have the same result on the cell that binding of AMPA or of glutamate to the kainate receptors has. And you need, for example, binding of AMPA, you need activation of AMPA and NMDA receptors before glutamate can activate kainate receptors. So you have a basically...
So there's no point in looking if they sort of complement the distribution? Because functionally they actually have...
Because they're not always found in the same layers, but it's interesting that NMDA receptors and AMPA receptors are both found at higher densities in the superficial layers than the deeper ones. And in kainate receptors, it's the opposite pattern. But then, of course, you've got the connectivity within an area which we haven't mentioned. When you go then to other receptors, then you find, for example, the M2 receptors have a very high density in primary auditory cortex. The alpha-1 receptors have a very low receptor. So you've got a higher receptor, a higher M2 density in TE1 than you have in TE2, but you've got a lower alpha-1 in TE1, TE2, and it gets higher as you move then towards the higher auditory areas, if you want to call them some way. And then, for example, the same thing happens with AMPA. It's lower in the primary than when you move up to TE3. Okay, now another part of the extended Wernicke complex is our areas 40, 39 of the inferior parietal lobule, and there's a study by Svenja Kaspers, and she studied the cytoarchitecture of these two areas, and what she found was that Brodmann hadn't done a good job here. So where he only described two areas, but where other researchers such as von Economo and Koskinas had found up to 7, 8 areas, she could also find a total of 7 areas. And the borders again were confirmed with the observer independent method. And if we then look at maximum probability maps of these inferior parietal areas, so these are coronal sections at 4 different rostrocaudal levels, we here have so TE1, TE3 in this level. Here we have it as well, and if you move back further enough, obviously where the lateral fissure moves up, and then well, Brodmann's area 22 would border with his area 40. We find borders between TE3, which would be a part of 22, and PF, OP, and with PF, which are part of Brodmann's area 40. Yeah. Okay. And again, looking at receptors, we will also be able to confirm the existence of all these subdivisions between Brodmann's areas 39 and 40. And again, not all receptors showed all the borders, but say, for example, this border here between area PF and PFT. It's found at the same, it's revealed at the same position by changes in the densities of AMPA receptors and kainate receptors and NMDA receptors, GABA B receptors. So it's basically shown by all of the receptors on this slide. Whereas I'm looking for somebody who doesn't show the border. Da, 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 da. Ah, yes, this one here. Svenja used a dotted line here. I don't know if you can see it on the slide. She used a dotted line to show the position at the border, which is revealed by the other receptors. So it means that here in this section we have PFOP here, and we have area OP, or we have an opercular area here, but the M2 receptors don't show that border, okay? Cytoarchitecture, well, again, we've got in all areas we have a relative strong what we call a columnar arrangement. So it looks as if the cells are arranged in nice straight lines that run perpendicular to the cortex. That's typical of the PF areas, not of the PF or of the PG area, sorry. Right, now I'd like to go into another question that we can answer with receptors, that we can ask with receptors, sorry, and that is, what do receptors tell us about hierarchical organization in the brain? And do they tell us something about possible functional relationships between the Broca and the Wernicke regions? And the reason why we think that we can answer this question is, when you look at a section like this one here, which shows the muscarinic M2 receptors in a coronal section through a whole human hemisphere, you'll see that there are some parts in red with a very, very high density of M2 receptors, and then you've got other areas with a very low density. And this little knob here you should recognize because we've just been talking about it. That's Heschl gyrus, right? So we've got the primary auditory cortex here, and this part here with a very high M2 receptor density is area 3B, so it's the primary somatosensory cortex. And the primary motor cortex has a very low M2 receptor density, and if you look at a section with the primary visual cortex, you'll see that the primary visual cortex also has a very, very high M2 receptor density, and it gets lower as you move into so V2, V3, V4 areas. So visual areas which are higher in the hierarchically higher visual areas. And this is something that holds true not just for the human brain, but also for the monkey brain. You can see, well, a little lump here. So this is a section, coronal section through the brain of a Macaca fascicularis. And with its little Heschl gyrus, with its central sulcus here, and with a very, very high M2 receptor density here, area 3B, and down here, area TE1, and a relatively low M2 receptor density in the primary motor cortex. So this made us think the densities of receptors are telling us something about the position of an area within a functional hierarchy, because no area works by itself to enable a given function. It's always a network of areas, okay? But the fact that this is much higher and then it gets lower as we move down here, it's telling us something about where this area is in the hierarchy. Is it at the beginning of a sensory input, or is it somewhere where it's integrating input from different sensory modalities? And of course, because a single neuron doesn't express a single receptor type for a single neurotransmitter system, but it can have input from, or it has receptors for loads of different neurotransmitters, and so different receptors for different neurotransmitter systems. That's why we decided to serially section the brains and to routinely examine as many different receptor types as possible. Now we're limited by the number of sections, but mainly by the existence of ligands which specifically label the receptor that we want to examine. And so routinely we examine, as I mentioned before, between 15 and 20 different receptors, and if you look at this, you can see this kind of pattern, and you see, so my M2 receptor here with a very high density, it actually also has a relatively high density of nicotinic receptors compared to other cortical layers. You see here, for example, that the cortex has a very low density of nicotinic receptors compared to the thalamus, and dopamine receptors are very low in the cortex compared to basal ganglia, for example, whereas the cortex, for example, has many more 5-HT1A receptors than subcortical structures have, and so on. So this gives us different ideas, or hopefully.
Sorry, is there a way to sort of, I think it's really a badly formulated or unformulated question, idea I have, because a key thing in the function of these areas, cells, layers, is cell types, right? So cell types will give sort of the different function of these things. What can we say about cell types using this kind of
With this method, nothing. Receptor autoradiography has the enormous advantage that what you're labeling is active functional receptors, right? So the tissue, it's not fixed, so it's alive. The receptor is functioning. It's the membrane-bound receptor. So what you're seeing are the receptors that would enable function in the living brain. That's the big advantage. The disadvantage is they could be located in the presynaptic membrane, they could be located in the postsynaptic membrane, and that would basically result in a different global activation of the area, okay? And of course they could be located on different kinds of cells. We don't know. The nice thing would be to not just compare receptors with cell body stainings, but then to go into immunohistochemistry and look at subunits, the problem is that many of these receptors are they're protein complexes. So if you're looking at the 5-HT1A receptor, which is labeled this section here, that's no problem because that's just one protein. So immunohistochemistry, no problem. But you're looking at where's my GABA here? My GABA A receptor. 5 different, it's not even 5 different proteins. It's a pentamer, it's a heteropentamer. You've got, the alphas and the betas and the deltas and the 1s and the 2s and the 3s. And when you do immunohistochemistry, you're not just labeling the proteins that are membrane bound, you're also labeling the ones that are somewhere within the cell on their way to the synapse or whatever, but it would be the complementary work to be able to do all of these things. The other problem is, to do this, we need unfixed tissue. If you want to do immunohistochemistry, good quality immunohistochemistry, you need fixed tissue. So then it gets difficult to be able to, but it would be.
I was just interested if you can use this to kind of say anything about cell types. And I wonder if I don't know if you probably can't use different sections labeling different sort of ligands, receptors, to try and match up.
What I do, if I wanted to look at subunits, is I wouldn't look at 20 different receptor types plus all their different subunits. I mean, you'd be, the number of structures that you want to look at then increases exponentially. I'd concentrate on a single receptor type and then look at all its different subunits. That's the thing that I'd do. Okay, well what we do here with this material that we have is, once we've got an area that we've identified, we can extract the receptor densities from that area, okay? And we've expressed them as fingerprints. And basically a fingerprint is a polar coordinate plot in which you've got so as many receptor types as you've examined, and that is the axis. So down here in the middle would be 0 receptors per femtomole, and up here we'd have 3,000. And then on each one of the axes, you plot the density of the receptor in that area. And what you have here is like a cloud of points, and it's difficult to see the pattern here. So what we do is we join the points with lines, and we get rid of the dots just to make the fingerprint nice and tidy. And now that's the fingerprint of TE1, okay? So the fact that these lines here in the middle, they don't mean anything. It's where the lines cross the axis that gives you the information. And what we found is that areas which we know are involved in different functions, they have different fingerprints. So here in green there's TE1, and in yellow is the fingerprint of Brodmann's area 17, which we know is a primary visual cortex. And that would be the fingerprint in blue of the primary motor cortex. So these fingerprints are giving us information about the relative balance of all these receptors within a given cortical area, or subcortical if you extract from a subcortical structure.
But through all the layers.
Well, in this case, it's through all the layers, but if you want to, you could do a laminar specific.
For some areas there were highly
Yes.
significant differences between.
Yes, we do both. We do both. I'm just showing the mean areal densities here, but yes, you could have fingerprints for layers 1, 2, 3, 4, 5, 6, or 3A, B, and so on. You can go into.
For serotonin, 5-HT, there was a huge difference between
Yeah.
red and blue. So
Because
if you put them together, you have some green.
Exactly. And the thing is, sometimes when you compare the fingerprints of two areas, of the mean areal fingerprints of two areas, you don't find any differences. But if you break them down into the layers, then you would see, there are very big differences, because in this area, all the receptors are in the superficial layers, and in this area, they're all in the deeper layers, but the mean is the same in both. Yes. That happens, yes. It's just the next level of analysis. Okay. So what we did was use these fingerprints to examine the relationship between different areas of a cognitive system, and we used the sentence comprehension related, or areas involved in sentence comprehension, and other brain regions. And this is a study in collaboration with Angela Friedrichi, and starting point of the study was 3 activation sites elicited by functional MRI. So we have a blob in a region which we've called IFS, IFJ, because it's located in the inferior frontal sulcus, in the inferior frontal sulcus, close to where it joins the precentral sulcus, and we know that we can find these areas in here, but this blob actually occupies several of these cytoarchitectonically defined areas. And then we have a functional blob in the posterior superior temporal gyrus, superior temporal sulcus region, which was an activation elicited by lexical syntactic integration. I didn't really, it's not that I didn't have enough space. The citations, the references for these studies are in this article here, and you'll be getting the slides anyway with the talk, so you can look those up. And then we have a third functional blob region, which we called Area 44D because it was located completely within cytoarchitectonical Area 44D. And how we did this from the technical point of view is that we actually have MR scans of the brains that we've processed for receptor autoradiography. So we could overlay the blobs on the MR scans of those brains and identify the sulcus and the gyrus on which the blob was located, and then that was where we took the measurements of the receptor densities. And then we used a series of areas which are cytoarchitectonically defined, and the color coding that I'm using here is red is areas that we know are somehow involved in language production or understanding. And we have a series of control areas which would not be involved in or non-language related areas. So there's the primary somatosensory cortex, area 3B, then there's the primary motor cortex and the primary visual cortex. And you see I've got area 4 twice because we measured receptor densities at 2 different locations within the primary motor cortex. One, a relatively ventral one, the position where the mouth representation is, and we know that part of the primary motor cortex is involved in at least spoken language. And then the other measurement is located dorsally. One could argue that that's also involved in language because it's where the hand representation is. So of course, if you use sign language, well, it's also language production. But the classical language-related area would be the spoken language. That's what one thinks of as language-related area. And then we have a selection of multimodal association areas scattered around. And I like the way we put area 46 as multimodal, non-language related. But we did. So frontal lobe area 9, 46, inferior parietal lobule, the selection of areas into which Brodmann's areas 39 and 40 could be subdivided. Area 7 from the superior parietal lobule. There's area 32 in the anterior cingulate cortex. And then here we've got areas FG1 and FG2. They are fusiform gyrus areas. And we know that they've been identified with the cell cytoarchitectonically. They've also been identified with receptors. And if you overlay the maximum probability maps with studies dealing with face recognition area, you see that I think it's FG2 is part of the face recognition area, but it could be FG1. I don't remember. One of them is definitely part of the face recognition area. Anyway, all these turquoise areas are so-called multimodal association areas. And then I
feel compelled to make a comment because we are a smallish workshop. There's some interest, and I feel a little not great about saying this because one of the co-authors is, I think of this paper, is making claims that I now don't understand. So if you go back one slide, here the claim is based on this particular organization that the syntactically relevant structure is area 44, the dorsal subdivision. Right?
Sorry,
So area 44, the dorsal subdivision,
yes,
is ostensibly the area associated with aspects of structural processing. Right? That's at least what's claimed in this paper. Now, yesterday I had the pleasure of looking at a poster here by this young gentleman who was reporting, I don't know your last name, sorry, who was reporting on a paper by Angela Federici in a functional activation paper, making the same claim, but crucial to that paper was that it's area 44, the ventral subdivision. In fact, not the dorsal subdivision for structural building. So I'm happy to believe both, but now there's a tension. What's it going to be?
The reference for this functional blob here is in here. I can look for it for you and give it to you afterwards if you want. We've heard this morning that lesions in different parts of all of these areas result in dysfunctions in different parts of speech production and understanding. So now maybe my keywords here, hierarchical syntax processing, are just too general, and it's not, now let me see. You see, here I write that 44V is also involved in hierarchical syntax processing. And there are a load of studies showing that as well. But the tasks that the volunteers were given are different in this study as maybe in these studies here. So maybe the dorsal and the ventral parts of Brodmann's area 44 are just involved in different aspects of syntax processing. It doesn't mean that the one blob doesn't exclude the other. But syntax processing is a very complex process. Could I say it that way? Would that be?
We have language comprehension, which is again something else. It's 47. It's 47. So, yeah.
Language.
There's a reason. I mean, the reason it's worth just kind of going down this path is now, for the first time, we have the really incredibly important, valuable exercise we're going through with you is to really be careful, even pedantic about the anatomic subroutines, and this is their opportunity for us, right?
You know,
we want to now be equivalently careful about how we map the cognitive science onto this, and not so.
When I move 2 slides on or 3 slides on, there's a comment about something like, look what I noticed preparing these slides, and what the reviewers actually, because this has been published, the reviewers didn't notice this, and we didn't notice it when we were writing it, and nobody noticed it, and I just, when I was preparing the slide, I thought like, so yes, it's it's a wonderful point, and I don't think it's a negative comment to make.
No, I mean, it's a challenge to us as a community,
yes.
right? I mean, so our homework to some extent is to say, look, now we know based on these incredibly important data how much more granular the organization is in an anatomic context. So we should actually rise to the occasion and say, let's be much more careful about how we subdivide this stuff.
Absolutely, and we have classified. It's here. We've classified area 46 as a non-language related area. They didn't notice this mistake here, and they didn't notice the mistake. I'll ask you later when I get to the slide if you notice the mistake that not even the reviewers saw, thank God. Okay? So yes, it is very challenging, and obviously, listening to the talks this morning, listening to you talk about the different parts that are affected and how by the different lesions and how the patients react, we should have examined. We should have had regions of interest all along here, different sites all along here. We can do that in a future study. This was a first attempt to see if transmitters can tell us something about the molecular structure of an area and the relationship between the molecular structure of that area and the functions that it's involved in. Now this slide here shows what we call, what at the Institute we've now dubbed stamps. These are just little cutouts from the different areas. So for example, this is the posterior superior temporal gyrus superior temporal sulcus, and it's just a narrow strip of the area in which up here at the top you've got the cortical surface, and down here you've got the border with the white matter. So then you can put your stamp collection, you can put them all next to each other, and you can compare the laminar structure of the different areas, in an easier manner. And so this is the AMPA receptor for different areas. There wasn't enough space on the one slide to show all the areas that we examined, and then you'd have the kinase and the NMDA, and what you also see here are we've identified the cortical layers, and you can see that they're not. So we've to make the figure simple, we've actually made all our stamps the same size, although we're aware of the fact that these different areas aren't equally thick, but the relative width of layers, then you can see the differences between this stamp. And then we went on to extract the receptor fingerprints for all of these areas, okay, and they're shown on this slide here. And so you've got the polar coordinate plots, the receptor densities or the receptors. No, okay, a spoiler. So the receptor order is the same for all the areas. What we see basically is always a bias towards very high GABAergic receptor densities, which is something that holds true for all areas, because GABA is the most predominant neurotransmitter in the brain, and it also has the highest, or GABA receptors have the higher densities. And we could spend hours saying, well, I think this fingerprint and this fingerprint are more similar to each other than this one, and so on. Oh, and these slides here, so the filled in surface is the receptor fingerprint itself, and the dashed lines show the standard deviation. Okay. And as I said, we could spend hours saying, oh, this looks more similar to this than to that, and so on, and oh, this is very different from everybody else. But we don't want any subjective comments here. We try to be as objective as possible. So what we did was transform these fingerprints into feature vectors. So basically, you've got for each area a a line which is given by all the different receptor densities, and you can feed this and you can do a hierarchical clustering analysis, which would then group objects which are more similar, and that's what we did here. Okay, this shows the result of a hierarchical cluster analysis of all the areas examined here. And we did it in the left hemisphere because it's the language dominant hemisphere. And if I bring in a bit of color to make it easier for us to see areas, you can see that all the areas which were labeled in red, so all the areas which we had identified as language relevant areas, are all located together here in one cluster. We've got area 46, which is down here, and it is a language-related area, but from the neurochemical point of view, it's more similar to, well...
I don't think it's a language-related area.
Sorry?
I don't think it's a language-related area. It's more executive control.
Okay.
946 are part of the executive control network, so I don't think 46 is claimed to be a language-related area.
But...
I agree. You're looking at me. I agree. I don't think so we looked at 46 in our patients, and patients with lesions in 46 do have trouble with more complex grammatical constructions that require working memory and executive functioning. And in our analysis that we were talking about, area 46 was really very, very small.
Okay.
7 was the more dominant one.
Okay.
And so
You're happy with this? Well, then I'm happy. Okay, we've got all our language-related areas. We've got multimodal association areas down here and up here, and then we've got our primary sensory areas here, and we've got the part of the primary motor cortex where the mouth representation is, with the language areas. So what area should be somewhere else and not where it is? What's the area that the...
Auditory, primary auditory.
Exactly. When I was preparing this slide, I thought, hold on a second, TE1 is basically, according to the model, is it Alice? I'm very bad with names. But the slide I showed with the, it's a review article on the areas that we now think of as part of extended Broca's and Wernicke's regions. They include area TE1, which of course you could argue against. You could say it's not the sole input of language, and
it's not surprising that it has a slightly different configuration than the other areas because they serve different functions, right? So it's kind of...
If we look at the primary auditory cortex, we normally like the type of functional contrast that would give activity there would be from silence versus noise, or any type of sound versus any other type of sound, and not specifically when you compare like different elements of language.
Yes.
Then you normally don't find the activity there, you find it in another place. So I would not consider it a language area, although without it we don't have language. But then, yeah.
Well, we'd have the sound language, but yes, we wouldn't have the.
What I would probably, I'm thinking about it, is the fusiform gyrus areas of the visual word form area. The way I think could be more involved in the language network.
Yes, but no, because of the word recognition area.
Yes.
The word recognition area is actually located a bit more rostrally than FG1 or FG2 are. So we've got new areas now. They're located there. There's FG3 and 4, and that's where but we haven't extracted receptor densities from those yet because we hadn't finished identifying them.
Can I ask one question? So I had in the past discussions with Katrien who said, well, that what we report language-related activation in 47 cannot be true, because if you look at the hierarchical cluster for the cytoarchitectonic relation between these areas, 44 and 45 are very similar, 47, so it must be frontal background. Now, so let's say from the cytoarchitectonic point of view, you get a totally different picture. Now we look at the receptors, and now it's
it's there.
47 is 44, are very even closer than 45.
You see,
so where are we now going to say? What are we going to believe here?
You believe the receptors. No, you believe the receptors not because I say so, but because you know the way I said that there's no function without structure. There's no function without receptors. So it is terrible to say, and I'm not saying that the cells are just like the scaffolding for receptors, but if you don't have given receptors, a cell can't react. So that's why I think receptors are giving us more information than differences in cell body distributions are giving us. So that's why I'd go for the. It's just, you know, you're going into more detail.
Mm-hmm. Okay, so I'm happy because we find 47 all the time, but then my conclusion is that the position has changed. 47 was considered to be in Jülich, not to be part of the.
No, and the other thing is as well, Katrien has never done a cluster analysis of cytoarchitecture profiles with all of these areas, which is of course also the next point. You know,
especially in relation to 44 and 45,
if you only have 44, 45, 47, 46, yes, so 47 might be more similar to 46 and so on than it is to 44 and 45, but in this global picture, no, it slots in there nicely. Okay? So we did yes?
Do we think the histochemical profile of these areas are going to inform more which areas are in the language network or not? Is it not more the function of these areas are actually going to be informative? Because it may be similar, but maybe similar for some other reasons.
So did the hen come before the egg or whatever? Is that?
We can talk about
that's again another philosophical. These receptor patterns, this specific molecular structure, means that the areas when receptors are released in the brain, and of course we are thinking when we argue like this that the same amount of a given neurotransmitter is released at the same concentration overall in the brain at the same time, which of course it isn't, but sort of like thinking that these areas are somehow functionally correlated, which we do find co-activation patterns. So maybe the fact that they have a more similar molecular structure explains why they're correlated in their function. The problem with receptors is, of course, it sounds wonderful. You can say, well, you know, activation of a glutamatergic receptor will result in an activation, and of a GABA receptor it will result in an inhibition, but it's not that easy, because, well, is the receptor located on a glutamatergic neuron or a GABAergic one? So what is the global result of this? We don't know. Oh, okay, well, I only have one more slide, and it's the reviewer's fault that we had this to make this slide, because they said, well, this is all wonderful, but what happens in the right hemisphere? And we said, well, it's not the language dominant one, so who cares what happens in the right hemisphere, you know, but we did it. Obviously, we had to. And if I bring the same colors into it as before, what we see here is the same areas, the same receptors, the same method, everything the same, but it's just the other hemisphere. And now we only find areas of the Broca region grouped together, right? Here. But we find these other areas, there's TE2 and area 47 and 4V and this posterior superior temporal gyrus, superior temporal sulcus. They are not in the right hemisphere. They are not related with the areas of the Broca region from the molecular point of view. And that's it.